Okay, so let's pick up where we left off. Oh my. And I will have a take home exam ready for you next Monday. So, and then you'll bring it back on Wednesday. So I don't think that'll be too awful. Although, oh, what the heck? Let me get rid of this thing. Put this over here. There we go. Okay. So we were in chapter 23 on slide 25. And it's, a lot here. In fact, I was looking over it and I was like, where in the world are we? Oh, get this thing out of here. There we go. Yeah, you guys can. Okay. All right. So we've been talking about how our hormones help control throughout an organism. And we talked about the classes of hormones, our paracrine and endocrine and all that good stuff in there. So let's briefly cover our major endocrine glands. So here we are releasing things through the bloodstream to have distance effectors. We have a few major ones that we classify in here, the hypothalamus and the pituitary, located in the brainstem. You got the thyroid below the throat and the parathyroids back there as well too. One we don't think about, our adipose tissue technically releases hormones that have system-wide effects as well too. You got your adrenal glands, your pancreas. I think your pancreas is insulin. Yeah, that's what we're going to be seeing there. And of course, our ovaries and testes, depending, will have some overall effects in there as well too. So we got a lot of sources of hormones, and we also have a fair amount of division of labor between the tissues. Okay. For the longest time, for most of my life, I'd say the brain is the most important organ, because it's where our center of consciousness is. It controls everything. It's the sequence in there. It's signaling everything else. But there's an argument to be made that the liver is actually perhaps the most important organ, because without the liver, the brain is going to die, because it synthesizes everything, and everything kind of goes through the liver to get other places in there as well too. If we look at our division of labor, we can look at the pancreas as well as secreting insulin and glucagon. And these are in response to various changes in blood sugar concentration. So if we have an influx of glucose, pancreas is going to say, hey, store this for later, use it for energy. If we're getting low, then it's going to release glucagon, say you're hungry in there as well too. Our portal vein attached, connecting to our liver here, is going to carry our nutrients that we digest into the liver. You got your intestines for digestion, your lymphatic system carrying lipids from the intestines to the liver. So a lot of things filter into the liver in there as well too. Adipose tissue is going to store and mobilize triglycerols. And we're also going to talk about thermogenesis a little bit here coming up as well too, where we can actually generate heat. And of course, your muscles are what allows you to do. So we all have all specialized organs in there. And these organs, if we can differentially regulate their function, that helps control or maintain the homeostasis of the organism. Now, we don't always want to maintain homeostasis, because you think of development, my son and his class are learning about reproduction and puberty right now, and they're all so, it's such an exciting time for kids to learn about, and I'm so mature learning about it, but yeah. But anytime you're going through developmental stages, growing into adulthood or a mature organism, you may see some non-homeostatic responses in there that are important for that growth. All right. If we look at our hormones, we can look at various target tissues, and I'm really not a big fan of slides like this one, because we see, everything just kind of feeds into everything else here. But let's take a look at our neuroendocrine origins here, coming from our central nervous system. So we basically get some type of input in here that's going to trigger a response. And so our hypothalamus is going to say, hey, let's release some hormones in here, release some factors. So our primary target, or our first target, is going to be the anterior pituitary gland. So the hypothalamus releases some factors that stimulate the anterior pituitary to release a series of hormones as well too. Whether we got corticotropin, thyrotropin, prolactin, oxytocin, whatever, all these will go on to second targets. So our Our first target is the pituitary. Our second target is maybe the adrenal cortex, or the thyroid, or the ovariotestories, or the pancreas, or whatever in here. And then those release other hormones which target multiple tissues. So I've come up with an analogy about why we have such a high level of regulation and so many steps in between everything. All right. So yesterday, I went to the zoo yesterday, right? It was a little cool in the morning, but not terribly cold, but it was a little bit cool in the morning. And then it warmed up. And then when the sun was out, it was really, really warm. But then if you're in the shade or when the clouds came over, there was a breeze that blew by that was kind of cold. So how do you make sure that you're at the right temperature so that you're comfortable? Wear layers, right? So my wife was making fun of me because, how many layers are you wearing? And like, well, I had on a thin t-shirt and then a shirt very similar to this one that was, was it not quite exercise material, but something like that. And then a very light jacket over top of that. And then, a hat as well too, which could either keep my head warm or keep the sun off my head. And the idea was that whole concept of layers. And you hear this as well too, like dressing for winter time, or and I'm sure you guys know coming into these buildings here, it's going to be somewhere between 56 and 86 degrees in here. So wearing layers really helps you maintain that, and you can adjust, take off a layer, add a layer, something like that, so on and so forth. So that's really what we're doing with all these. We have all these thin little layers that by themselves aren't very warm, or if you remove one, they aren't very cooling in effect. But if you start combining them, okay, you can get multiple effects in there as well too. Okay, so I don't know how good of an analogy that is, but I kind of like that because here's our first layer, here's our second layer, here's our third layer, so on and so forth. I like this jacket that I'm wearing right now because it's warm enough. It allows me to stay fairly warm, but this thing is wind permeable, so I've got a windbreaker to go over that as well too. And I'm like, yeah, how many layers are you wearing? So when you're studying this, you can you can say, how many layers of regulation are there? A lot, but it allows for that very fine control of homeostasis in there. Okay, or in response to whatever our environment's throwing at us as well too. There's a couple other ways we can look at our hormone signaling as well too. We can talk top down. Okay, some signals go from the brain, literally the top, and go out to the rest of the body. Things like oxytocin, vasopressin, cortisol. We also have bottom up signals as well too. They can go from someplace else and send messages to the brain. For example, epinephrine, your adrenaline getting on there, or our insulin coming from the pancreas, which is going to have neural effects as well too. And we'll talk about leptin here in a minute as well too, with some really cute mice. All right. Okay. So our hypothalamus is kind of the coordination center. I don't know if you guys saw that movie. I know you've got kids, so maybe you what was that? Is Pixar movie where it's like the emotion centers in there, and you got anger, and I can't remember the name of the movie now too, but yeah, it's sort of like that's our hypothalamus in there too. It's controlling everything in there. Like this, a small region in the forebrain in animals with skulls. Okay, but it kind of does a lot of signal integration from the central nervous system to synthesize these hormones, oxytocin, vasopressin, and a bunch of factors that also regulate the pituitary gland as well too. Okay, if we look at our pituitary gland, okay, a pituitary will release hormones that will target other glands as well too. And you've got your posterior pituitary and your anterior pituitary as well too. Okay. And really, I guess the major difference in here is short peptide hormones versus long peptide hormones, and the various targets that they can function as well too. So, rough sketch of them, of the pituitary gland here, in the hypothalamus. Pardon me. Whew, underneath the hypothalamus and all that good stuff in here. I always kind of look at these pictures and take them with a grain of salt. We're not trying to teach neuroanatomy here. I will say at one point I picked up a neuroanatomy textbook and said, I'm going to learn neuroanatomy. And it's hard. You got these lovely little pictures here to help out, but if you're actually looking at pieces of the brain, it takes practice. It takes a lot of practice. I look at neurosurgeon and I'm like, okay, and then, oftentimes the borders kind of fuzz over. I think I mentioned a girl I dated when I was in grad school had a brain tumor. And they used the gamma knife to excise the brain tumor, but the tumor was growing close to the motor coordination sites. So in targeting it, it's like she had difficulty with her left leg. Little needed some physical therapy for a couple years, and always had like just a little bit of a limp in the left leg because they had to the tumor was growing into them. It was a glioblastoma, but it was impinging on the motor cortex, so it was hard to remove it without um, you know, doing some damage to the motor cortex as well too. And
is she okay now?
no, she died.
Oh,
Yeah, I mean, she survived for like 4 or 5 years, but the glioblastoma they thought had been growing for a long period of time, just very very slowly. But the issue is after they removed it, it came back, and so then she went through like chemo and stuff like that, and then I think had another round of surgery, and it was constantly growing back, so they couldn't quite take care of it. But it wasn't a fast growing cancer. Like I knew someone else who had a different type of brain cancer, and it was like from the diagnosis to the surgery was about 3 days. For my girlfriend, it was a month and a half or something like that. So, but ultimately, it was pernicious enough that I mean, she had like a 4 year survival rate, which is pretty good. But yeah, yeah, but yeah, not all outcomes are good. It was actually interesting because so one of the symptoms that the reason we found it is because she went into a seizure, and had seizures from the impinging tumor into the motor cortex. Um, but she actually had enough therapy, enough surgery, because of course when you start having seizures, you're not allowed to drive anymore. But after about 2 years, she was actually allowed to drive, and bought a car and was driving and working and stuff like that. And then the tumor kind of came back, and it's like that Canadian animated short, The Cat Came Back. The tumor came back. So sorry, we have a horrible sense of humor in there as well too. Anyway, sorry to bring you down with that, but yeah. Not immediately deadly.
No,
Yes.
I was thinking about when you say it makes her home, like you broke up with a girl with the brain tumor.
Yeah, exactly. It's like, what? Your brain's not working. You're out of here. No, I'm cold. That's why you guys are all getting D's. D minus. No, sorry.
God, he's not joking about that.
I am joking. Yeah. But yeah, we all had a very dark sense of humor. One of the things that we did after, like a group of friends, we had a party for her after the first surgery, and one of the games we played was pin the tumor on the brain.
Oh my gosh.
Yeah, I mean,
You guys do have a dark sense.
yeah. But anyway, yeah, you gotta find ways to laugh at life, because anyway. If we look at some of our pituitary hormones, oxytocin and vasopressin, on a more positive note, oxytocin is the love hormone. It's also what's responsible for new baby smell. But it's the idea that oxytocin, one of the things it does is it helps induce the contraction of smooth muscles around the uterus during labor, and it also stimulates the release of milk from the mammary glands as well too. So that's why it's associated with new babies. The new baby smells to stimulate the production of milk, so that the mother and the child can bond, and the mother will want to spend the time to feed the baby. There are studies in animals, and I believe there have been a few cases of people who have an issue with oxytocin either not being produced or the receptors not activating in its presence, and they don't bond with people. One of the things, oxytocin, they call it the love hormone as well too. It's supposed to make people bond and care about each other. So if we could artificially induce that, maybe you could treat post-traumatic stress disorder or antisocial disorder or something like that. But those are just kind of out there right now. I will say that oxytocin also functions as a pheromone. That's basically part of our transmission of the from the baby to the mother. So it's at least the first known, if not the only known, human pheromone out there. If we look at vasopressin, completely different function here. Here we are looking at water reabsorption in the kidneys. I don't know if you guys have like a Red Bull habit or something like that, but there was a, you know, my wife is apparently trying to, scare my son straight, scared straight. I don't know if you remember those TV shows or something like that. But there was a story recently about a kid in the UK who'd been drinking like 4 energy drinks a day for 2 or 3 years and died of kidney failure because you just overload your kidneys with a lot of stuff that's in there. So vasopressin works for that water reabsorption if you're not getting enough water and instead you're drinking way too much bad stuff for you. Then you're going to see some negative effects on the kidneys. So don't drink like a half gallon of Red Bull a day. It's bad. Okay, it's bad. Oh my Lord. I'm old because I'm doing George Bush the first impressions. Both hormones together do function in pair bonding, but again, these are likely not from the pituitary. Again, oxytocin functions as a pheromone in here. If we look at these two hormones, they're actually really similar in structure. You can see the only differences are this isoleucine versus phenylalanine. That's a pretty big difference. They're both hydrophobic, but you got a nice ring structure over here. And here we have a leucine versus an arginine. Again, that's a huge difference in those two amino acids. So 2 small differences. Well, 2 amino acid changes, big effects in there. Okay, but it's kind of neat that they both have that disulfide bond with the cysteines in there. If we look at cortisol, so anytime that you have a stress response, you can see the production and release of cortisol. This can be fear, infection, hypoglycemia, and in basically any type of stress in here. But you will get this lovely little electrical signal in the central nervous system, which will stimulate the hypothalamus to reduce nanogram level amounts of corticotropin releasing hormone, or CRH. Okay, that goes to the anterior pituitary. It will allow this to release microgram amounts of corticotropin, which will then go to the adrenal gland, and you'll release milligram amounts of cortisol, which will go and have its downstream effects on the muscle, the liver, and adipose tissue as well too. Okay. So if you think of, we were at the zoo and we happened to walk past a couple who were sitting on the steps, and one person was eating. It's like, yeah, anytime I get stressed out, and it's just so stress eating is a thing, and cortisol can promote that as well too through its effects. I want to say on the adipose, but I'm not 100% certain that that's well known at this point. But the main key here is, we see this initial stimulus releasing nanogram, then microgram, then milligram amounts. So we have this, if we go back to signal transduction, all the way back to chapter 11, I believe, that signal response, that what's the word I'm looking for here? Enhancement. That's not quite it, but you get that magnitude of response in there. So small amount of initial stimulus results in a massive response to it. And of course, you can see that our cortisol will go back and inhibit the releases as well too, because we want to have adaptation. But if you constantly have high levels of cortisol, then you're going to see inflammatory responses and all sorts of stuff starting to add up. And this may play a factor in various diseases as well too, possibly even in immune. But I'm getting ahead of things we don't need to know. All right. In terms of our bottom-up signaling, we have to have tissue-specific responses to various fuels that we have out there. Okay. So I know we've talked about importing glucose and then transporting glucose to, say, the muscles or the heart or the brain. So if we look at the portal vein here, it's going to carry our nutrients into the liver. Our various liver cells will turn these nutrients into fuel. Okay. And then our liver enzymes tend to turn over very quickly compared to other protein enzymes that are out there. And these can change with diet and the needs of various other tissues as well too. So for example, what we got here? Yeah, this is just kind of an overview. We're going to talk about ghrelin and leptin coming up here in a minute, as well as adiponectin as well a little bit. but we'll see that, our AMP kinase pathway in here is going to play some effects. Basically, these will come from either our adipose tissue, our stomach, or our intestine. And you can see the stomach and adipose tissue will send signals to the brain, which will release other compounds in here. You can see from our pancreas, these can also kind of feed back here as well too. We can either have high glucose levels or low glucose levels that will increase insulin or glucagon, and have various other effects there as well too. Hey, they didn't do a glucagon. Anyway, this is just kind of a nice little summary slide. And we're gonna get some more details about this. So our liver is kind of our most important organ because it's going to be adapting to our changing metabolic conditions. I get hungry, the liver is going to send something in there. I get full, the liver is going to send something in there. Okay, so what we need is a way to sense and process various nutrients in the liver and then signal the other organs, primarily the brain, about those sensing and processing activities. All right, so we can see our glucose, fructose, galactose, mannose, all those are going to feed into and get converted into glucose 6-phosphate. All right, and our glucose 6-phosphate, as we talked about, we saw this right at the start of the semester. It's kind of our transfer in the liver. We can go into the glycolytic pathway. We can go into the pentose phosphate pathway. We can export it as fuel. We can make glycogen. So there's lots of them. It's kind of like our grand central station there. In our liver cells, we have the GLUT2 transporter. And one of the crazy things about this is the GLUT2 transporter lets glucose go in and lets glucose go out. It's going to go either way. And it's really just kind of a concentration gradient in here. We also have this hexokinase 4 in there as well too, which we sort of talked about a little bit before as well. It tends to have a higher Km than other hexokinases, so it is going to bind glucose 6-phosphate and make it. But if the glucose concentrations are fairly low, it's not going to bind it as much. All right, so that glucose, recall that once the glucose is phosphorylated, it's stuck in there. It's like the crazy cat lady who puts the little shock collars on the cat, so once they come in their house, they can't get out. Well, in the liver, she doesn't get all the cats collared up. So if the cats are wandering around in the house and some new ones wander in, they're free to wander back out. Okay, same thing with our glucose here as well too. The neat thing about this too is hexokinase 4 is not inhibited by glucose 6-phosphate, so it's constantly making a low level of glucose 6-phosphate, but it allows glucose to go back out into the bloodstream where it can get transported to other tissues as well too. All right, so suppose that we actually do make our glucose 6-phosphate. What do we get? Actually, all these little minor details in here should be capital G, 6, capital P. It's bugging me every place I see it, but it's not that. We can dephosphorylate it to give us free glucose to send to other tissues. We can make it into glycogen. We can send down the glycolytic pathway. We can go into glycolysis to make ATP or triacylglycerides. And we can go into that pentose phosphate pathway. All right, so this is just kind of like a little visual representation of that. Our fates of glucose. And again, you've kind of all seen this before, all right? So we're going to look at, so that's our glucose. Same thing for amino acids. They can have various functions in there as well too. So if I eat some protein, I can recombine amino acids into proteins for the liver. I can use them, like we saw in the previous chapter, as precursors for various hormones and nucleotides, or I can feed them into the citric acid cycle. Okay, so we can, and we can also make pyruvate, which we use for gluconeogenesis, or we can convert into acetyl-CoA for energy for the liver cells. So if we need more glucose for other cells, we'll shunt that into the gluconeogenic pathway. If the liver needs more energy, we can shunt that into the acetyl-CoA in there. Or if we are well fed, we can make lipids out of that as well too. Fatty acids in the liver, we can make liver lipids as well too. So the liver can take care of itself, or it can oxidize these as fuel to make energy for the liver, and then we can send excess acetyl-CoA as ketone bodies to the brain and heart. So if you undergo the Atkins diet or restrict your carbohydrates, you're going to be primarily using ketone bodies. Some acetyl-CoA we saw could be used to make cholesterol as well. We can convert fatty acids into phospholipids. We can convert them into triacylglycerides for storage. And of course, we can send our fatty acids to the heart and the muscle for oxidation. All right, so really 4 and 5 are related. It just depends. If we send them to the heart and muscle, they're going to get oxidized. If we send them to the adipose tissue, they're going to be stored for later. All right, and again, we can see visually kind of what's going on in here as well too. Okay, so a lot of this chapter is review, and this is a long chapter, so I'm trying not to, I'll have a better idea on Wednesday of what your exam is going to look like for next Monday. So don't sweat all this too much here. Just make sure you say, okay, so this is what happens to carbohydrates in the liver. This is what happens to proteins in the liver. This is what happens to fatty acids in the liver. And it's going to be the regulation that is going to be the most important in here, because how do we know?
The entire exam going to be take home?
Yes.
So on
Yeah.
Monday, we're just going to jump into the next chapter?
Yep. Absolutely. Yep. Actually, maybe even on Wednesday. So, because we don't have much time left.
When is our last exam?
I want to say May 12th or 13th or something like that. It's like the second week in May, I believe. But I'll look that up and find out for sure. Acetyl-CoA, we can make our ketone bodies, and the neat thing is, if we go on to prolonged fasting, or we don't go on a diet that we have no carbohydrates, these ketone bodies can give 70% of the energy for the heart during prolonged fasting. They can also be utilized by the brain as well. Okay, let's talk about fat. We talk about our adipose tissue, and there's actually 2 types of adipose tissue. Most commonly, we think of the white adipose tissue, which is where we just kind of store triacylglycerides, and that's where we're storing excess energy there as well too. When we think of fat, that's kind of what we have there too. But there's also brown adipose tissue, which we don't tend to see as much of. It's much more present in, say, like newborns or in various other animal species like rats. We'll have a lot as well there. But there's a neat thing. You can actually interconvert white adipose and brown adipose tissue depending on environmental exposure. The white adipose tissue tends to be larger and has like one big droplet of fat in there, where our brown adipose tissue is a bit smaller, and it kind of segregates the fat into several droplets in there as well too. They also tend to have more mitochondria, and it gives it kind of this brownish color, which is why we have named them. So here's our fat. Our white adipose looks white. That mitochondria tends to give it this brownish color. That's why we call it brown adipose in there too. All right, so why do we have 2 types of adipose tissue? Well, our white adipose tissue is fuel storage. That's basically a big old warehouse. You go out, out towards Kernersville and see like the Amazon distribution center in there too. That's like 1 million square feet. Boom, that's our white adipose tissue. We're just going to store everything here and send out fatty acids as we need to. On the other hand, the brown adipose has a different role. It has this uncoupling protein, UCP1, also known as thermogenin, which allows the proton gradient in the mitochondria to be dissipated, so that instead of actually making ATP, you generate heat. So thermogenin, for thermogenesis, we actually produce heat in there. So in a low temperature environment, this will serve to keep the organs warm. Okay, so we often find our brown adipose in newborns. Even as adults, we have it, but it's primarily expressed around the central organs here, and I don't think we have it really much in the brain, right? Hold on here.
Is the visceral fat that they talk about, is that white adipose tissue?
I'm not sure.
They say is the most dangerous one.
Yes, yeah. Yeah, that would be white adipose. Yeah. Because the neat thing about brown adipose is you're basically burning that fat to produce heat. So you can actually interconvert that. So I don't know, there's like cryotherapy or something like that. So if you spend a lot of time in cold environments, you can convert white adipose to brown adipose, and you will start expressing uncoupling protein and burning fat as heat. I've often wondered, so where I did my graduate work, there was another department that was looking at particularly they were interested in fMRI studies of the brain, and they looked at Tibetan monks. And they talked about you can take a Tibetan monk in a freezing mountain monastery, unheated, wrap him in a wet towel, and an hour later the towel would be dry because he generates so much body heat. And I wonder, they talk about is that meditative potential, or is that just a high conversion of white adipose to brown adipose, so that they're generating a body heat through thermogen in there as well too. Um, Typically, when a child is born, they'll have a large amount of brown adipose because they've been inside the womb, so now they have to keep warm. So a lot of the fat that they have will keep those internal organs warm as they adapt to the atmosphere outside. As they grow and mature, brown adipose declines and gets converted into white adipose. But even in adults, spend a lot of time in the cold. Um, you can convert that white adipose to brown adipose. So go hang out in the freezer if you want to. I don't know.
Now I wonder what that cold therapy is now that they're talking about. That they melt the fat with cold therapy.
I wonder if that's I have to look. I'm always hesitant because I think when I was in grad school is when that one commercial started for it's like I'm a doctor, like hydroxycut. And it's literally, hey, you lose 20 pounds in a week. But it's, yeah. But it's all water weight, and you're not really actually, getting rid of any fat or anything like that. You're just, removing a lot of water. So I'm skeptical about anything, but I would have to look into whatever that is up in there as well. The cold therapy in there. But this is not going to be in any immediate thing as well too. I always think one of the jobs I had in between I worked in a food line deli, and so I had to go into the cold room there as well and get the wings out or whatever. And so I was like, well, let's spend more time in here. I don't know. Same thing in grad school. I worked in a cold room for a while, and whew, man, it's like purifying a protein. It would take hours, if not over a day, for this thing to cycle through multiple things in there too. And I got to the point where like, well, let's work through the window so I can stay warm out there.
Yeah, I don't know if the whole cold thing works because I was overweight even when I was working. My first job, I cleaned the chickens at Walmart and seasoned them in a freezer.
Oh,
it was freezing.
Oh,
So I think if the cold made that much of a difference on the.
I was gonna say I was definitely a bad employee at Food Line because whenever I go in there, like a lot of people loved the wings that we had, and they were actually pretty tasty. So when people order a dozen wings, it's like I get 13 out, 12 for you, 1 for me. So it's bad. It's bad. I'm not built for retail. All right. Hormones tend to work actually really quickly on adipocytes, and this can be accelerated by epinephrine. Epinephrine will give a boost to the hormone-sensitive lipase to access the liquid droplets so that they can free up free fatty acids for energy. On the other hand, insulin is going to inhibit this. And again, epinephrine is going to say, hey, we need energy, we need it now. Insulin is like a signal that now we got lots of energy, we can store it for later. Okay. If we look at muscle, we got 2 types, our slow twitch and our fast twitch. Slow twitch tend to be fed by a lot of blood vessels, and they're very rich in mitochondria. So they provide energy through slow and steady oxidative phosphorylation. All right, so that's kind of maintaining things in there too. Our fast twitch have less mitochondria and lower oxygen delivery, but they use up our ATP a lot faster. You get a lot more fatigue in there. So I look at, I'm a hockey fan. I've been watching hockey games there too. And it's like, they do like 30 to 45 second shifts. I'm like, how do you get that tired in 30 to 45 seconds? But using a lot of fast twitch muscle in there. You got the endurance training to increase the amount of mitochondria in there so you go further. But then you watch a hockey player who's been on the ice for like a minute and a half, and they're struggling. They're struggling. And I'm like, yeah. And I think about that when I played basketball too. I got my story about aging out and how fast twitch muscle declines with age. I played a lot of basketball in my 20s, way too much basketball, and I played with guys who had nicknames like Fingers and Magic and stuff like that. So I learned a lot. I actually got decent. Yeah, it's good. Where people who didn't know me, I'd play with them, be like, oh yeah. I definitely didn't look like much. So I moved down here as a professor, and I was taking my son for swimming classes over at Wake Forest, and at the gym there would be these kids playing basketball. And when I say kids, I mean college students. So 19, 20 years old, and they sucked. they were awful. I mean, they're terrible. So I'm like, well, yeah, I need some exercise. Well, my son's swimming. I'll go and I'll play basketball with these guys. And here's the thing. I had talent. I could pass. I could play D. It's like, yeah, my shot was off. It'd been a long time, but that's okay. I could do all the other stuff in there too. But after 10, 15 minutes, I'm like, huffing and puffing up and down the court, and they're all young and running up and down. Like, they couldn't make a shot, couldn't pass, couldn't play. They couldn't do anything. But I had the skill set, but I couldn't do anything because I was so out of shape at that point. So I definitely did some more endurance training to get back in shape for that. Of course, maybe I should cheat and take like erythropoietin or EPO enhancing drugs to be a better athlete. You can think back if I don't know if anybody ever follows cycling. It's like, I have no interest in cycling whatsoever, but the Tour de France, it's just fun to watch him crash in the mountains where it's so pretty. But was it Lance Armstrong who had all his titles stripped from him for performance enhancing, basically doping? And it's, you look at all the doping agencies. Was it the Russians in the last Olympics? And it's like, we're going to suspend the entire Russian sport federation because the doping was so endemic. It's a constant race of what can you do to enhance the edge versus how can you catch that? And it's fascinating because a lot of times some of the things that you can do, like you look at the hormones, steroids, and the athletes who have shrunken testes and greater risk of cardiovascular disease as well as aggressive behavior and all sorts of stuff. Is this worth it? What's the payoff, risk versus reward? You go back to the 90s and all, it's just doping was crazy. I always think of Barry Bonds. I grew up watching the Pittsburgh Pirates, and I remember when he was a skinny guy, and then all of a sudden he's huge in like 2 years. I'm like, okay, yeah, that's completely natural. On the other hand, now you got to look at what you can do to your children too. It's like, hey, let's give our kids human growth hormone and make them bigger, stronger, faster. It's a trade-off, understanding and abuse, and then unseen downstream effects as well too, because everything's interrelated. That's definitely one thing you should take away from this class. Everything is interrelated. All right, if we look at energy sources for our muscles, typically we're going to be using in our activity, we're going to use phosphocreatine. We're going to generate from our glucose, make our phosphoenolpyruvate, make phosphocreatine, and you get heavy activity, and that's going to be come out there for our quick, fast activity in there. Fatty acids, ketone bodies, blood glucose for our lesser activity. We also see muscle glycogen that gets used up again for heavy activity as well too. And our muscle glycogen producing the glucose, that's going to give us lactic acid. So if you feel the burn, that burn is lactic acid building up in your muscles because you can't clear it fast enough back to the liver and convert it into pyruvate or glucose or whatever. But that's the feeling, the burn there too. Interesting thing about muscle glycogen, liver glycogen varies wildly. Muscle glycogen, you are going to, short burst of muscle glycogen, but as soon as you're done, you're going to start regenerating that muscle glycogen. It's kind of fascinating. It's kind of like our adipocytes, where something like 70% of the fatty acid turnover, the hormone sensitive lipase cleaves it into free fatty acids and glycerol, and 70% of that gets immediately converted back into triacylglycerides. Your muscle glycogen is maintained at a near constant concentration, whereas liver glycogen is all over the place. Okay. All right. Sources of ATP for the skeletal muscle glycogen in there. Kind of mentioned that. Phosphocreatine. Our creatine is a much higher energy compound than ATP, so you can use phosphocreatine to produce ATP. Okay. If you look at kind of a neat little thing that goes on here, during exercise, you can, you'll see a spike of our inorganic phosphate, and then you'll see what is this spike in phosphocreatine. Then you'll see our ATP levels over time in here. So you have our what is it, rest, exercise, and exertion. Our phosphocreatine is being used to make ATP to try and keep these levels constant even during exercise. And you can see as our phosphocreatine is utilized, we see an increase in inorganic phosphate that gets released as well too. So our axis is really kind of like the z-axis going into the board here. Look at that. Okay, so let's talk briefly about glycogen. All right, how do we control glycogen mobilization? We sort of mentioned this before. We're kind of summing this up. Epinephrine is one of these, and it's very similar to glucagon. But our epinephrine, we can bind that to our G protein coupled receptor, and we will activate our G protein, which will result in the formation of cyclic AMP from our adenylyl cyclase. Okay, but one molecule of epinephrine can activate about 10 G proteins, which will stimulate the release of about 200 molecules of cyclic AMP, which will activate our protein kinase alpha, which will activate phosphorylase B kinase, which will activate our glycogen phosphorylase, which will, we'll see our production of glucose 1-phosphate. So basically one molecule of this can stimulate the release of 100,000 molecules of glucose. So it doesn't take a lot. If you look at epinephrine, boom, fight or flight, you've got to have that energy. There is a massive amount of energy released right away. I think this also helps, in our comparison with, say, glycogen to amylose and amylopectin, starches found in plants for their energy. Our glycogen has all those ends, so you can cleave off lots of glucoses very quickly from our various enzymes in there. You got lots of ends that are free, so you can release lots of glucose, as opposed to a potato. If you scare a potato, it's not going to try and run away. It doesn't need a lot of energy. It's going to try and grow or repair the damage. So you can look at the structure helping aid the function of the various biomolecules as well too. All right, so what happens if you exercise a whole bunch? Like me playing basketball with the college kids, just hunch over and breathe a whole lot, okay? So we have what's called an oxygen debt in there. So even after the exercise ends, we're gonna see this need for oxygen to help rebuild the proton gradient and replenish the ATP levels that are used there. So we can use ATP for gluconeogenesis, so that we can use up the lactic acid that's been released in the muscles, and we can restore that muscle glycogen concentration. This is going to happen through the Cori cycle. So here we have our muscle glycogen releases glucose, it makes ATP, and we have lactate as our lactic acid as our waste product. This gets released into the blood, sent to the liver, where the lactic acid can be used in gluconeogenesis to make glucose, which can get transferred back to the muscle to make to restore the muscle glycogen. All right, so that's our Cori cycle. I want to make jokes about that, but I'm not gonna.
Make the joke.
Well, it involves our former dean who is not here. Dean Walker's first name was Corey, and I was like, oh, the Cori cycle. Well, time to go to another school, and
No.
except I just find it entertaining that he's the only administrator I know who, when they left, didn't have a party.
Oh,
Yeah. I don't know why that would be, but clearly not one of my favorite people. But that's okay. There's a very good reason for that, because I'm, in his words, the white devil.
the what?
Because
He called me the white devil. Yeah, indirectly, but I'm like, yeah. I'm like, do people actually use that term? I thought that was, an anachronism left over.
White people in high school would say it, because that's
Yeah,
something like a Beartron would say.
Yeah. It's like, it's like the man. I remember teasing my grad school roommate about being the man, because he was the only one of us who wasn't technically some form of minority or disadvantaged group, because he was a white guy from upstate New York. And I was like, you're the man that's been keeping all of us down. But not serious. But apparently some people still use the white devil serious thing. So anyway, so
Yeah, I used to
got
say
them
when I
all.
was trolling in high school. I got in trouble for it though.
Yeah, it's and it it must be tough. I'm about to have a teenager, so you think of the things that kids do and they did, and the things that you said and the way you interacted with your friends and within your groups and the people who knew you. And then of course now you have social media, so you can take all that out of context and just post something on there. It's like, oh, this is terrible. I look at my extended family. I keep waiting for someone to accuse me of cultural appropriation with my various Indonesian batiks that I wear. Which there's actually a whole nother line there too, because I've learned a whole lot about the Malaysian Indonesian conflict too. And it's like, oh, yeah, but it's funny because all these, well, almost all but one or two have come from my extended family. So they're like, here, yeah, we want you to wear this. We want you to have this. We want you to, but no one ever does because I'm too boring and dull. It's kind of like when I taught the class about, the chemistry of sex and drugs. And I couldn't even get on a list of horrible college classes. So it's like no one pays attention to me. I don't know. I was trying. I named it as provocatively as I could, which was funny because it was always full. There was always a wait list for it for the name. But anyway, let's jump back in here. Our heart muscle versus our skeletal muscle. Okay. So our muscles, we use them a lot and then we rest and use them and a lot of rest, but our heart muscle is pretty much always on. So it's got a lot more mitochondria. Mitochondria is about half of the cell volume in there. And it's fueled primarily by fatty acids as well too. We can use some ketone bodies, some glucose, some phosphocreatine, but mainly we're burning fat in there as well too, which is another reason why I have to be concerned about cardiovascular disease, and high cholesterol and all that good stuff, affecting the heart function as well. It's a very aerobic organ. If you cut off oxygen to the heart, like any muscle, it will die. That's where you get our myocardial infarction, all that good stuff in there as well too. If we look at the heart muscle in here, you can see our contractile apparatus is kind of sandwiched in between mitochondria. So we're providing energy, so you can have this constant contraction. I don't know enough about heart function to go into much detail, but I do know that your was it aortic, blips in the aortic function are on the sorry apical, is that the right one? As opposed to ventricular. Ventricular mishaps or miscues are bad in a case, and generally predict something bad is going to happen, like myocardial infarction. Whereas on the other side of the heart, that's not as bad. There can be blips on that side, and there are different chambers in there. Random things that you pick up. If we look at the brain, our brain is also very oxygen dependent as well too. It will consume about 20% of oxygen in the body at rest. So if you're thinking, even more, right? So typically we need about 130 grams of glucose a day. So right here I've got my brain fuel, which has 46 sugars in it. So I just need to drink like 3 or 4 Mountain Dews a day. No, three Mountain Dews a day. Well, actually, sometimes I did drink about 3 Mountain Dews a day, but that's also when I started getting developing my hara, as I say, for Aikido. But but the brain is highly dependent on glucose. There are cultural references to brain candy, but quite literally, sugar is good for the brain, and but not excessively. That's what the brain wants. Okay. And of course, we're going to make lots of ATP in there to maintain electrical potential. So we can look at, in particular, our sodium, potassium ATPases. Also, calcium plays a major role in there as well too. But these are all interconnected as well. So fuels for the brain: just glucose and ketone bodies. No fatty acids in the brain. All right. We can look at our blood. I don't know if you guys ever sold plasma for extra money. I remember as an undergrad, there was was it Ceratec, some little company that would pay you for your blood plasma that was located literally not even a quarter of a block off campus. It wasn't quite like right across the street from the campus, but it was 3 buildings down. And that's where people would go buy beer money. Terrible. But what's in there? You got your erythrocytes and you got your water, your various cells in there, leukocytes, platelets as well too. and your plasma is primarily water, and then you got a bunch of solutes in there, basically various metabolites and waste products. You got to carry our urea and uric acid to the liver and kidneys for disposal. You got lots of proteins in there as well too. Again, our isotonic solution, if you ever go to the hospital, you get an IV, right? Where they give you, 0.9% saline solution. Why is it 0.9 saline solution? Because that is isotonic for the bloodstream. You don't want it to be hypertonic or hypotonic, or else your blood cells will swell and burst, or they'll shrink up and crenulate, and either way, you get loss of function in there. Same way for people who need to be fed, they give them 5% glucose intravenously, because again, that is a isotonic solution for feeding. All right, what happens if we change, if we have low blood glucose in humans? All right, so our normal range, resting somewhere between 70 and 100 milligrams per 100 milliliters in here. We start dropping below that just a little bit, and you'll see the release of glucagon, possibly epinephrine. Cortisol is a stress in here, maybe a little bit of sweating or trembling in there. But you get hungry. All right, blood sugar drops, you get a little bit hungry. Drops a little bit further, you kind of get that little jitters, a little shakes in there too. Yeah, it's like, okay, it's really low blood glucose. Let me go get a candy bar or something like that, and you're all well and good. Drop below 40 milligrams per 100 milliliters, and you start getting really tired in there too. And you can see convulsions and coma. And if you keep on going lower and lower in blood glucose, you will see brain damage and ultimately death. So we got to maintain our blood glucose at a certain level. Good news is, for most people, low blood glucose is not typically an issue. It's usually high blood glucose. You see that in diabetes a lot in there. But ideally, you want to keep this around 60 to 90 milligrams per deciliter. Okay. Come on. Yes. Maybe. There we go. All right. Insulin is going to stimulate the conversion of excess glucose to glycogen or triacylglycerides as needed in there. So what we see is insulin stimulates glucose uptake in the muscle and fat cells, right? So not so much in the liver, because the liver is constantly bringing it in and constantly releasing it. But for our muscle and fat cells, it's going to say bring it in, all right? In the liver, insulin is going to stimulate glycogen synthase so that the glucose that does come into the liver will get converted into glycogen in the liver glycogen here. We can also see that it's going to stimulate fatty acid synthesis so that we get our acetyl-CoA, and we can make fatty acids, and then we can export those through VLDL and send them to our fat tissues for storage, or to our other organs for usage as well too. In our adipocytes, insulin is going to stimulate triacylglycerol assembly. So we're going to take those free fatty acids and glycerol and store them for later. Okay. So here's a good overview of like the previous 7-8 chapters. And here's more, because this is really a big summary chapter in here. This is all the stuff you should know by heart now, right? Okay. All right. So we can look at the organism in a couple of different ways. And one of the ways to look at this is a well-fed, maybe a postprandial organism, as opposed to a starved state. So fed state versus starved state. So if we look at our well-fed lipogenic liver that we have here, what we see is there's lots of glucose that comes into the liver, so we can make glycogen. We can go convert it into pyruvate through our glycolysis to get our acetyl-CoA. So we can make ATP that way. We can use acetyl-CoA to make triacylglycerides. We can send those to adipocytes for storage. Our VLDLs can also carry them to the fatty acids for use as energy as well there. Our intestine, I should have started over here and said our glucose comes into our liver. And we're going to see, our insulin gets released here as well too, to stimulate a lot of this as well. So this is our well-fed state. We're exporting energy, basically. We're setting it to be stored or to be used in the brains or muscles. Okay. If we look at our beta cells in the pancreas, they're the ones that are going to secrete insulin. We almost never talk about the alpha cells or the delta cells in here, but our alpha cells secrete glucagon. All right, so which basically has the opposite effects of insulin. So insulin, well-fed state; glucagon, not well-fed state, starved state. So it's going to stimulate the opposite pathways in here. And then our somatostatin has other effects as well too. I love the little cartoons that are in here, because there's just so try and compare that to an actual slice. But anyway, our glucose regulation of insulin secretion. Okay, so insulin modulates glucose uptake, but glucose modulates insulin secretion. All right, so if we look at our pancreas, our glucose enters our bloodstream. Right, it's going to enter the beta cells through a GLUT2 transporter, where we'll see it'll be phosphorylated, go into glycolysis. We'll see our increased levels in ATPs. As our ATP concentration increases, it'll bind to a potassium channel. Those will close, they'll depolarize the membrane, and that will trigger the opening of these calcium gated channels. So we'll see an influx of calcium. Calcium concentration will increase, and we will see insulin that will be released from these secretory granules where they're stored after they're made. Recall that we started with the pre-proinsulin to get to our proinsulin, and then our mature insulin stored in secretory vesicles, which can be released as our glucose levels increase. Okay, so again, we have this separation of function. Our pancreas brings in glucose, which goes into the glycolytic pathway, increases ATP, stops the potassium gated channel, stimulates the opening of the calcium gated channel, which the influx of calcium stimulates the release of the insulin vesicles from where they're secreted, and then insulin can go to the other organs and have its effects. Okay, not like which include enhancing glucose uptake in other organs. Okay. If we look at our ATP-gated potassium channels, they're often the targets of various sulfonylurea drugs. So sulfonylurea drugs are an old treatment for various diseases, including diabetes. So if we look at these sulfonylurea receptor in here, these drugs can close the potassium channel, and they artificially stimulate insulin secretion. So in this case, we have a type 2 diabetes here. So we overcome the resistance to insulin by just increasing the insulin concentration that's circulating. So we might not have enough insulin in there to have its downstream effects. So if we add more insulin, then by adding more of our substrate, it's more likely to bind to its receptor and trigger its effects in here. Okay, so it's not the best way, because if you keep increasing the concentration of insulin, you're going to keep enhancing the insulin resistance. But it was a reasonably effective for at least early stage type 2 diabetes in here. Okay, this is a mechanism of how it works. I am not going to ask you guys the mechanism and all that good stuff in there as well too. Okay, glucagon has effects that are kind of opposite of insulin. Okay, so it's going to work in a cyclic AMP mediated cascades. In the liver, it will activate glycogen phosphorylase and inactivate glycogen synthase, basically the opposite of insulin, right? In the liver, it's going to reduce the amount of F26BP, fructose 2,6 bisphosphatase, so that it inhibits glycolysis and stimulates gluconeogenesis. Again, the opposite of insulin, as you're bringing in all this glucose and say, hey, now we have a lack of glucose, so we want it to go the opposite direction here. Glucagon also inhibits pyruvate kinase. It leads to the accumulation of phosphoenolpyruvate. It's going to stimulate PepsiK, our phosphoenolpyruvate carboxykinase, to stimulate gluconeogenesis as well too. Right? Glucagon has effects in the adipose tissue. It is going to basically tell the adipose tissue, don't use glucose, so that you can send that glucose to the brain. So our glucagon is going to say we got to keep the brain functioning. Our adipocytes, even though they do play more roles than we tend to think, other than just energy storage, they are less important than the brain. Okay, so we see that fatty acid transport goes to other tissues, and glucose can go to the brain. Okay, so if we look at our starved state liver, our fasting glucogenic liver, okay, we don't have anything coming in from our portal vein or anything in there as well. So what we see is as we're doing activity, we kind of break down proteins, and as the breakdown, they get sent back to the liver for metabolism. You can see we can have our various amino acids, which can get broken down into, say, pyruvate, go through gluconeogenesis to make glucose, which can be sent to the brain. We can also see various ketone bodies that get made from these as well too. Those can be sent to the brain. You can see our adipose tissue releasing fatty acids, goes to the liver. Our fatty acids get converted into ketone bodies. So again, we are sending primarily glucose and ketone bodies to the brain, and our fatty acids to other places. Ketone bodies can go to the muscles as well too. Right? So a little bit different function here, kind of the opposite pathway. All right. So what happens to blood glucose levels when we have insulin in here? We see glucose uptake increases in the muscle and the adipose and the liver. Glycogen synthesis increases in the liver and the muscle. Glycogen breakdown decreases in the liver and the muscle. We see glycolysis and acetyl-CoA production increase in the liver and the muscle. Fatty acid synthesis increases in the liver, and triacylglyceride synthesis increases in the adipose tissue. All right, so these are various targets that are in here. So glycine. So let's compare epinephrine versus glucagon. They both have very similar effects in here. Okay, they both stimulate the same cyclic AMP signal cascade, but one of the neat things about epinephrine is it also stimulates glucagon secretion too, and it inhibits insulin secretion. Epinephrine also breaks down muscle glycogen as well as liver glycogen. So normally, even if you're a little hungry, your muscles are going to maintain their glycogen. But epinephrine is going to say, mobilize all the energy now. It's that fight or flight response. Okay, so that's also going to result in the stimulation of glycolysis in the muscles, and we're going to see more lactic acid formation as well too. You guys ever I always think of the example a couple years ago, like right after I got my car, and I was driving through an intersection, and it was 2 lanes that I was in, and apparently the car coming the opposite direction didn't see me behind the car that was turning or something like that. I was going through the intersection, so he started to turn across right in front of me, and I had my kids with me. I just bought this car, and I was like, and I'm sold. This could be my car commercial, because I swerved out of his way because there's no chance to stop, and I missed him by about 6 inches on that side, and then the cars that were waiting to turn on one of the other streets intersection, I missed them by about 8 inches on that side and got through there. But that's a close call, and so bam. Epinephrine release, right? What happens a few minutes after that epinephrine release, after everything's settled down? You get that crash, right? So that's that lactic acid filing through and rebuilding that liver glycogen and all that stuff in there as well too. So that's one of our big differences between epinephrine and glucagon. You get that little crash in there. I'm going to pause for a second. So I got a BMW. It was a used BMW, mind you, but a very nice used BMW. But it always makes me think of the best car commercial I ever saw for BMW. One of the reasons I wanted a BMW since I was younger, back in my college days, is we used to watch Cops. I think you guys remember Cops, and like World's Wildest Police Chases and all that stuff in there as well too. And of course, all those shows, they always catch the bad guy, right? Almost all the time, like maybe like 2 or 3 times a season, somebody will get away. So there was a kid, 18-year-old guy, who stole a BMW, and he was running from the cops. And so he at one point takes an exit and is going uphill around a curve on this exit, and there was a police car, and these were like the Ford Tauruses that were souped up at that stage. That was a little bit closer to the intersection, was trying to cut off the intersection before he got there. He accelerated uphill around the curve and beat the police car to the intersection. I was like, all right. There's another point too where he's driving and the police car pulls up beside him, and he does a head fake and a little wheel thing like that, like he's gonna turn into the police car, and police car does a little swerve there too and loses control and spins out, and he's fine. He just drives away and recovers like nothing. I'm like, I'm looking at this, and he actually got away and he was arrested like 24 hours later or whatever, because but I remember looking at that, I was like, well, if I ever need to outrun the cops, I better have a BMW. Yes. It's terrible, terrible the thoughts that go through your mind there. Okay. So, but anyway, that was my adrenaline story there too, of avoiding an accident. All right. So let's spend the rest of today talking about how we actually use our fuel over a metabolic time period. So I don't know, maybe you guys are a little more normal than me. Actually, today I actually did have breakfast. So ate breakfast. You wake up at a starve state, eat breakfast. You get fuel use over there. It kind of tails off over 4 hours, and then lunchtime I should eat lunch or something like that, right? So what we see is immediately after a meal, our glucose levels increase. We're taking in nutrients in there. We're going to see insulin being released and stimulating glycolysis and glycogen synthesis. A couple hours after we finish eating, those blood glucose levels start to tail off there as well. We begin secreting glycogen to make up for that, and the liver glycogen will release glucose to maintain those blood glucose levels. After about 4 hours, our blood glucose levels are going to start to decrease, and we're going to see the release of glucagon. We're also going to see some more triacylglyceride hydrolysis to provide energy that way, and fatty acids start to become fuels for muscle and the liver as well too. Okay. If we fast for longer than 4 hours, so you can think of like overnight or something like that, or I don't know, some of you may have had like doctor's test, something like that. Don't eat anything for 24 hours. You fast for 24 hours for this blood test, or if you ever had like your lipid tests, fast overnight, don't eat any breakfast or anything like that. So if we have prolonged fasting, and we start to actually use the muscle tissue for fuels as well too. So the liver, again, we can go through that whole deamination, transamination concept that we talked about in the previous chapter in there. Our carbon skeletons of our glucogenic amino acids will get converted to pyruvate, and then glucose and our ketogenic ones can make our ketone bodies so that they can get exported to the brain, heart, muscle for energy as well too. What we also tend to see over time is that the blood pH will decrease as well too. So we'll start to see some ketoacidosis, okay? And that's why, like if you suddenly go on the Atkins diet and cut out all carbs, you can have this massive drop in blood pH, which can stimulate some really bad effects. So you kind of want to ease into any major dietary changes generally. All right. So the other neat thing is we see some similarities between prolonged fasting and type 1 diabetes in here. Okay. So basically our type 1 diabetes is, we are not seeing any insulin production as well too. So we tend to have lower blood glucose. All right. So we start off with protein degradation from the muscle cells, giving us our glucogenic amino acids. Those go to the liver, and we produce a lot of urea. Okay, so we've got a lot of nitrogen in there, our transaminations, deaminations, all that good stuff as well too. We also get our oxaloacetate, which can feed into the citric acid cycle, make our phosphoenolpyruvate, make glucose. So our glucose gets exported to the brain primarily. We can also see, if we start running low on oxaloacetate, we have excess acetyl-CoA, so we can start making our ketone bodies. Our ketone bodies can get released and sent to the brain as well too. Excess ketone bodies will end up in the urine. So that's why sometimes, say diabetic, when they have very low blood sugar or prolonged fasting, your breath may smell like acetone. Your urine can also smell like acetone, but usually not too much in there. We see our fatty acids. They start, releasing our free fatty acids into the liver here. Fatty acids get converted into acetyl-CoA. Eventually, we run low on oxaloacetate. We get to that fatty acids in here. So let's look at what happens over a week. Okay, we see our blood glucose levels drop a bit here, plasma concentration, okay, millimolar concentration, but our body is trying really hard to keep that glucose at a constant level. So that's maybe a 20% drop, 30% drop in here. It's not optimal, but that's enough glucose for the brain to run. All right, so we're okay there. You also see that our our ketone bodies start accumulating. Beta hydroxybutyrate really amps up there as well too. Acetoacetate and acetone not quite as much. You also see the release of more free fatty acids, but again, this kind of slightly much like it kind of mirrors glucose in a way. As our glucose levels drop, our free fatty acids increase, but it's not as dramatic as our beta hydroxybutyrate that we see in here. All right, let's take a look here at cortisol a little bit. We talked about our cortisol being produced ultimately as kind of like our third step in here. So we get milligram levels of cortisol that are released. So we can think of cortisol as a stress hormone, but fear, injury, and all sorts of things can result in this. Pain, anxiety, infection, low blood glucose, so on can stimulate the production of cortisol. So one of the interesting things about cortisol is it can change the type and levels of enzymes. So we might see different isoforms, we might see different concentrations in there. So it doesn't actually activate or deactivate existing enzymes, but it changes their type. So in fat cells, this tends to increase fatty acid and glycerol release. Okay, so if you're high strung, you might be like pacing or shaking or fidgeting or something like that. That tends to require energy, so you see that release in there. We're also going to see in the liver, it's going to stimulate Pepsi K for gluconeogenesis, so that you can make more glucose and release that. And the muscle is going to break down protein for more gluconeogenesis as well too. Cortisol can counteract the effects of insulin. If you have a constant level of cortisol, remember, it kind of feeds back and inhibits the hypothalamus and pituitary glands in there. But we see adaptation over time. If you have a constantly high levels of circulating cortisol, then its effects are lessened. You may actually compromise the ability of your body to make cortisol, and ultimately it can lead to bone damage, muscle damage. The immune system can be weakened. Basically, other hormone functions can have negative effects from a constantly high circulating level of cortisol in there. I always used to mix this up with cortisone, and you get the cortisone injection in your elbow, so they have for anti-inflammatory in there. It's like, no, cortisol is different. So, but cortisol is our stress hormone. In response to an immediate stimulus, it's a good thing. It helps counteract that stimulus too, but you do not want to have constantly high levels of this because the immune system function there as well too. So if you're constantly stressed out, you're more likely to be sick, right? So more likely to catch diseases or something. So we should all do yoga in that. Okay. We will pick up on Wednesday with the diabetes, or as Wilfred Brimley would say, diabetes. Okay, good. That's not too old now, too. Yeah, I remember Quaker Oats. I remember he used to be on like some NBC Sunday night family show. I can't remember what it was. Our House, or I don't remember. But I remember growing up with Wilfred Brimley, and then he started selling Quaker Oats, and it's like diabetes. Okay, but we'll pick up with that on Wednesday. Finish up this chapter on Wednesday. And I'll go over what you'll see on the exam on Wednesday as well, too. There's this out here.